This topic is not directly on theme, but it's maybe interesting to people, and it's what I've been working on the last few months. So I'd like to talk about it. It's the most exciting thing in my head at the moment. In one sense, everything comes from somewhere else in some sense. And one of the motivations for this work was a very interesting paper which appeared 5 or 6 years ago in PNAS, where these people, Micheletti, Di Stefano and Orlan, said that there were very few, if any, knotted RNA structures known, in contrast to proteins and certainly in contrast to a lot of DNA. So I started thinking, well, I've been working long-term with Miff Evans, who'll be talking later, on a big project on trying to understand tangling in general. So I sort of ducked off to have a bit of a break from that project and thought, let's look at this. So I've been trying to understand what sort of tangles can you get if you have strands which self-assemble simply due to complementary base pairs. So a double helix, which we can just call a duplex. So a duplex strand is where we have strands which are self-assembling just by self-complementarity. And so the number of base pairs is dictating the twist of the helix, essentially. So the simplest tangle we could have, I'm sure everyone's familiar with, this is just a picture I took from Wikipedia, a cartoon of the standard form of DNA discovered by Watson and Crick. And the idea there is that you have complementary nucleotides in DNA, where the hydrogen bonding is what pulls the strands together. And so you have C, depending on the nucleotides in DNA, you have C complementary to G and A complementary to T. In RNA, you have very similar picture. This is a picture actually taken from, in fact, surprisingly recent, surprisingly rare crystal structure of RNA, which is similar to DNA, not exactly the same, but similar, because there are chemical differences. But essentially well, I'll describe a little bit more later, but basically it's the same idea that you have duplexing due to complementarity, except the thymine nucleotide in DNA is swapped for uracil in RNA, but otherwise you just have this complementary nucleotide. Now, the point that's interesting about RNA, and this is the ssRNA in my title, SS stands for single-stranded, is that RNA will often fold onto itself. So a single strand of RNA can have self-complementary, can have complementary nucleotides, so it will spontaneously wrap up onto itself. And that's an interesting problem, and in some sense, it's a simpler problem to solve. So here's a cartoon, again taken off the web, off Wikipedia, I think, where we're just following a single-stranded RNA. And conventionally, biologists talk about, because DNA and RNA are polar, so they have directionality due to the chemical structure along the chains, you start at the 5' end and you go along, you orient it so that you go from 5' to the 3' end. And if you look at this example, for example, we start at 5', we're heading through a region here which we're just going to call a single-stranded region because it's not duplexed. Then we head into a duplexed region because this bit of the strand is linking up with another complementary bit later on. So it's single strand, double strand, then it comes around, forms a hairpin loop. Then it's forming the other strand, local strand in the double helix. So that's also the same duplex, and then exits with a single strand. So if we take that whole string and just pull it out and look at what's going on, as we run along the string from the 5' to the 3' end, we go single strand, duplex, hairpin loop, duplex, single strand, and then 3'. And the duplex here is just being induced by these hydrogen bonding, so the complementary base pairs. And the essential point here is that the innermost nucleotides at one end of the duplex are bonded and the outermost are bonded. So you have a rainbow type arrangement. So we're going to simplify that whole picture topologically to just a single arc. So we're going to condense all these arcs collapse them all into one single arc. And we're going to capture that very simple fold topology, which is essentially, for my purposes, is just a single duplex by a single arc. So that's what I'll just call a linear ribbon diagram. So we have and I'm going to call this an annular ribbon. So the way to read this diagram in the linear fold is if we see a single arc an untwisted arc, we know that that's a single duplex. And then we can close that up, and that's more convenient for our purposes. So close up the linear arc, and then we just have an arc spanning two points, spanning, it's just some sort of chord spanning that circular diagram, circular perimeter. So the circular perimeter, the way to read this diagram is, as you move along a chain, you're actually walking around the perimeter. And every time you run into a ribbon, you're running into a duplexed region, then you've gone back to a single-stranded region, and then you're running into its complementary duplexed region. So that's the way to read this circular diagram. And the point about that diagram is that arc is actually, I'll show you in a minute, is actually describing an anti-parallel duplex. So that's a very, very simple fold topology, but we know in biology that RNA forms much more complex topology. So here's a picture that is taken off a fairly recent paper, which looks at the RNA genome in dengue fever virus. Okay, so if we just look at the top half, for example, or the bottom, look at the bottom half, is probably easier. That's the secondary structure. So here we go, we're starting at the 5' end, and as we move along towards the 3' end, we've got these non-local pairings, so these arcs. And these arcs are all describing, or nearly all describing, duplexes in this case. So we know that in the biological system, it's a very, very complicated fold. And what I really want to understand is, how can we start to classify these sorts of more complex folds way beyond a single double helix? And can we sort them, and can we come up with arguments to decide which one would be most likely in biology, and so on? And then the other question, which goes back to the first slide, is which of those folds are going to be genuine knots? In what case is the strand going to be knotted, and when is it not going to be knotted? But my classification is really got to do with the duplexing. It really is more general. It's more specific, if you like, than just the knot problem. So we can have different folds which give us the same knot, but we're still going to classify them as distinct folds because they have distinct arrangements of duplexes. So there's a real biological system. We haven't got that far yet, but let's see how far we can go in a very simple theoretical analysis. So first of all, I'm going to assume the string is closed because that just makes everything easier, for example, to understand knots for obvious reasons. We can define them rigorously and so on. So here's one way of closing up a pair of duplexes. So we have a duplex on top, or here's a strand. Let's just do it, take it carefully. Let's start at A, and we just walk around this strand. So we start at A, we go through a duplex, then we come out at B, then we go through a larger duplex, come out at C, go back through the same larger duplex, D, then back through the smaller duplex and back to A. So A, B, C, D, A, that cyclic walk is characterized by this diagram, where now instead of a fat ribbon, I've split the ribbon up into rulings. So in this case, I've put 3 rulings on the green ribbon, because the green ribbon is describing that duplex, which has 1, 2, 3 crossings, so 3 half twists, and the red duplex has 2 crossings. Okay, so that diagram describes this fold, which is now just a pair of duplexes in series. Now we know that a twist in principle of a double helix could be right-handed or could be left-handed. So we're going to allow for both. So in this case, we're going to call plus twist a right-handed twist. It's just a convention. So let's call that +3 and +2. So each of those ribbons has a twist associated with it now. And that characterizes the number of turns in the double helix. So each ribbon is a double helix, and the twist associated with that ribbon is the number of turns in the double helix. Now we could get a more general, a slightly more complicated problem would be, instead of 2 in series, we might have 3 in series. And if you just follow this walk around again, so we start A, follow it round, A, B, and then we go all the way back down here, and we come back to C, then we go back to A. So A, B, C, A. What's going on is we're going through the blue helix. So as we start at A, we walk around and we come through the blue helix, then we go through the red helix, then we go through the green helix, and then we do our hairpin turn here. We come straight back through the green. So we go it's the combinatorics that I'm trying to give you a feeling for here. So we're just going blue, red, green, green, red, blue. So we're going blue, red, green, then we do a turn, then green, red, blue.
Stephen,
so that ribbon diagram on the right is describing. Hello?
Can I ask a question about this?
Sure, sure.
The way you draw the knot is over, under, over, under, over, under. But it seems to me that if I make a change at one of the crossings, different over, under, it doesn't change your ribbon diagram. So it's not a one-to-one correspondence between the two.
Where do you want to change the crossing?
So for example, in the picture that you show right now, the middle twist.
Yes, the red one.
Yeah, take the middle crossing.
What the middle? You mean the middle duplex or the actual crossing?
The middle crossing. And
so
change.
Do you mean this one?
No.
Is this what you're calling a crossing? Sorry, I don't understand it. But...
A crossing is when one goes over the other.
So is that what you're calling the middle crossing, that one?
No, I don't call it the middle. That's the upper crossing of this.
That one?
That one, yeah. So
yeah.
now, so suppose I change it, that it used to be like this, now I change it to this.
Well, in my language, that will mean you're inserting a -1 twist duplex in there. Okay.
Where's the -1 here?
If you change the crossing from, you're talking about over, under to under, over.
Yeah.
Yeah, that will be a new diagram.
Yeah, but okay. Your ribbon, your diagram on the right doesn't change.
Yes, it does. If you change your crossing, you're changing my diagram.
How?
Because you're changing your crossing. I'm not talking about the entanglement. I'm talking about the actual.
But
if you change from an over and under to an under over, you're changing from a +1 to a -1 twist.
Yeah, but your diagram on the right doesn't change anything.
Yes, it does, because I'm adding a new duplex. Perhaps it's easier if I go with some more examples and then ask. Feel free to stop me again. Can I go a little bit further, and then maybe if it's still a problem, we can get back to it.
Sure.
I'm really not trying to dismiss you. It's not clear to me whether it's an issue interpreting what I'm doing or whether there's a genuine issue here.
Okay.
Is that okay?
Yeah.
Thanks. So a second way of closing up a single duplex would be, instead of closing it up with this hairpin loop, we could bring it from top to bottom in this sense. And for people who are familiar, this is very similar to different closures of Conway tangle. And now we have a completely different diagram, because as we walk around A, B, C, D, E, F, what's going on is we're going through again red, blue, green. So we're going red, blue, green, then red, blue, green again. So we go A, red to B, blue to C, green to D. Then we come back around, then we're going to red again, then blue again, then red. So you see the colouring is just cyclically translated around the perimeter in this case. And the difference there is really because what's going on is, in the first case, which is on my right here, sorry, these are anti-parallel. So if I just direct my strand, you can see that all my helices, the complementary strands, are oppositely polarized. And what that means is that, for example, if you start in this large central duplex, if you start with green, then you come out with red, then you go around and back, you come back to red, then go out at green. So it's green, red, red, green. Whereas in this case, it's a parallel duplex. And so it's red, green, red, green. So what I need to do is take my ribbon, instead of it being an annular ribbon, I need to twist it and make it a Mobius ribbon. So a Mobius ribbon, which I'm drawing with just in a fan type structure, is characterizing a parallel duplex, and the annular ribbon is characterizing an anti-parallel duplex. And every time I have a crossing, I need to define the sign of the crossing by the axis of the duplex. And this is a really important point now. The axis of the duplex is very well defined in RNA and DNA, because you have our base pair rungs. It's not defined in a knot, because I can have a +1 or a -1 when it's a single crossing. But in this case, it's rigorously defined in RNA and DNA. So you need to read these diagrams every time you see an annular ribbon, that's encoding an anti-parallel duplex. Every time you see a twisted Mobius ribbon, that's encoding a parallel duplex. Now, the next thing I'm going to do is just to come up with a sensible catalogue is we're going to get rid of a lot of what I'll just call the geometric details and collapse it in a sense to give me, I'll call it topology. It's a slightly loose word. It's somewhere between geometry and topology, but let's just call it topology. So the first rule I'm going to say is that if I have a whole series of duplexes, in this case, these are right-handed, 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 +1, +5, sorry, right-handed, right-handed, left-handed. So that's +1, +1, -3. So that's +1 twist, that's +5 twist, that's -3 twist. I'm just going to collapse them all down and sum the twists. So just collapse those 3 ribbons down to a single ribbon of twist 3. So does that help you anymore, Herbert? Does that explain anything, or it's still not clarified?
No, it doesn't. It doesn't really address my issue.
Can we perhaps? Okay. Do you want to wrestle with it now, or can we wait till the end?
Up to you.
I'd rather wait and get through it because it may clarify itself. It may not. I mean, I'm listening to you, but I won't. Maybe perhaps if we get more information, it will be easier.
Okay.
So thank you. Thanks for your patience, but don't go away. So the first collapse rule was our anti-parallel duplexes, which are just nested annular ribbons. Second collapse is when I have parallel duplexes. They're now distributed in a fan-like way around the whole perimeter. So here I've split my ribbons into rulings, and the convention I've used, don't worry about it too much, is that when I have an odd number of rulings, that's distinguishing an odd number of twists. And when I have an even number, so when I have three rulings, it's describing, in this case, it's a Mobius. So 3 rulings would be a parallel duplex with an odd number of twists. And if I had 2 rulings, it would be a parallel duplex with an even number of twists. Because the point is, it's really just the parity which is important for our purposes. Whether the number of twists is odd or even is what's important, because that determines which side, as my strand comes in, does it come out this side, or does it come out this side or this side? So it's the parity that's important, what I call the twist parity, even or odd. So there's my first rule, that if I have annular ribbons, if I can, you can think of them as sliding around the perimeter, I can condense them all together into one ribbon where I sum the twists. And if I have fans, I can close up the fan into a single fan where again I sum the twists. A third rule, which we can take or leave, but I'm just using it for convenience because it means you can get rid of a lot of fairly silly cases, is that if any time I have what I'll call just a free annular ribbon, so if I have an annular ribbon, which this is a free one, it's not intersected by anything else, and it's got nothing between its 2 feet, I can just slide it off and throw it away. So I just contract that to this, which is just the empty, what I'm just going to call the trivial fold. Okay, so really what that means is that this picture is something like this picture down the bottom. So really what it means is any time I have a fold, of course, any of these folds, I can take a little bit of my strand out and just twist it up locally. And a local twist like that just corresponds to sticking a small arc anywhere in the figure. So we're going to throw it away because there's an infinite number of those you could put in anyway. And you could just think of those as defects in a sense. So let's get rid of them. Now, the rule about this contraction operation, because this contraction is really throwing away quite a lot of structure, but it's giving you an essential topology. There's one rule, which is that I can always contract my ribbons as long as there's nothing in the way. So this is all free in between these ribbons. So those 3 ribbons can all condense down, and that whole diagram can collapse to this diagram. Okay, so there's a fan, a pair of Mobius ribbons, so they can close down to a single Mobius ribbon. But these ones I can't do anything with, you see. For example, the red and the blue are both Mobius ribbons. So there's a fan of Mobius ribbons crossed like that, but I can't close them up because if I try to collapse that blue onto the red, this green ribbon is in the way. So I can't contract these 2. So that's already contracted, that diagram. So the idea is that I can draw any ribbon diagram I want, which has any combination of Mobius or annular ribbons spanning the perimeter in any way, and that will describe a duplex fold. Of course, it may describe an uninteresting fold and so on. But what's interesting is that if I use these contraction rules, I'm going to get what I'm going to call a contracted fold. And every one of these, I mean, I should say that I now have an algebraic label to describe these folds just to make it convenient, but I'm going to refer in general to the contracted fold label because that's the one that's most interesting. So for example, this diagram here has got 1, 2, 3, 4 ribbons. So I'm going to give it a label which has 4 indices, and I've got 2 flags. So there's a flag, there's a fold label L, and there's 2 flags, a superscript, what I call the orientation flag, and a subscript parity flag. So the bare fold label is really just the combinatorics of the cyclic walk around the perimeter. So in this case, for example, ribbon 1, ribbon 2, so green is 1, red is 2, blue is 3, and teal is 4. So this walk is 1, 2, 1, 3, 2, 3, 4, 4. But of course, there's many different combinations of that because that ribbon label is completely arbitrary. So I could take any labeling of ribbons I wanted, and I'm just going to choose the one which has the smallest number, bare number. So in this case, it's 1, 2, 1, 3, 2, 3, 4, 4. So that's what I'm going to call the bare fold label. So that's the fold stripped of any information about the twists on the ribbon or the type of the ribbon. But now we put a superscript on it, which is telling us about the type of the ribbon. So the idea is that if it's a Mobius ribbon, which is characterizing a parallel duplex, we're just going to put an overbar on it, a sort of minus superscript. Okay, and that's because ribbon 2, which is here, is a Mobius ribbon. Ribbon 3 is a Mobius ribbon, and ribbon 4 is a Mobius ribbon. Ribbon 1 is an annular ribbon, so we don't put anything on it. So that's our superscript level. So 1 is bare, so that's annular, Mobius, Mobius, Mobius. And I only label the first occurrence. Every index, of course, occurs twice because it's double, I meet it twice around the perimeter. So I'm just going to label the first mention of that ribbon for convenience. Okay, so that's 1, -2, -3, -4. And now we have the subscript, which is this parity flag, which is just telling me whether I have an even or odd number of twists in my helix. So this label means that ribbon 1 has an odd number of twists. It's annular ribbon, so that's anti-parallel duplex, odd number of twists. Parallel duplex, odd number of twists. Parallel duplex, even twists. Parallel, odd, and so on. So this is a nice way of looking at it because basically what it tells you is that any digit string, I can throw down any digit string where every digit occurs twice, is going to describe in fact, a very likely uncontracted but a possible bare fold. So I can throw together any digits I want. And so that means that I can order them, for example, from smallest to greatest. So for example, if I have 0 ribbons, there's only one fold, which is that trivial empty fold. If I have 1 ribbon, there's only 1 fold. 2 ribbons, there's 2 folds, and so on. Up to 5 ribbons, there's 79 folds. And this is the smallest label. This is the largest label. So that's describing the walk around the perimeter, remember, 1, 1, 2, 2. So that label, for example, is uncontracted. 1, 1, 2, 2, 3, 3, 4, 4, 5, 5. It's describing exactly the fold where I just have 5 little arcs around the perimeter, which in my contraction, I remove. So that's going to be an uncontracted fold, for example. Now, the next rule is once I've got any of these digit strings, I can add any combination of orientation flags. So I can take my arc and I can make it a Mobius or an annular absolutely arbitrarily. Whatever I want to do is possible. So I can put in my Mobius annular flag, so in other words, overlines anywhere I want along these ribbons. Oh, sorry. The next rule is that I can also make the parity of any one of those ribbons even or odd. So I start with any possible label. I can have any possible combination of annular Mobius and any possible combination of even or odd. And any one of those labels is, in fact, I can draw it physically as a fold. But the point is that if I use my contraction rules an equivalent fold, and this is my definition of an equivalent fold, it will be equivalent if and only if it has an identical canonical label. So the canonical label is where I've looked at all my clockwise and anti-clockwise walks and turned my whole diagram upside down, and I minimize the digit string. This bare label L is the smallest number. So if you take a label and give it if you take a fold and give it a canonical label and write down that fold, take another one, if they got the same label, they're the same fold. So what do I mean by them being the same fold? What I really mean is that if a label, fully flagged label, so that means it has orientation and twist parities and annular Mobius flags, corresponds in fact to what I'll call a polarized rigid vertex strand graph. So for example, here's the fold, here's the rigid vertex strand graph. So what do I mean by that? What I really mean is that I'm going to take it's a degree 4 graph, and I'm going to take every vertex of the graph, and that vertex actually represents a double helix. Okay, so I'm going to collapse every double helix, every duplex down into a vertex. So all of these edges of the graph correspond to single strands in the fold, unduplexed regions of the fold. But at every vertex, I have to put a polarization vector through it. It's a degree 4 graph, so I can have a horizontal or a vertical, the way it's drawn here. So every second. So for example, this arrow, it's north-south or south-north. Think of it as I call it a plumb line. That plumb line is describing the axis of the helix. Okay, so you can read this graph. It needs to be a rigid vertex graph. In other words, the edge ordering is completely defined by the duplex type, and the polarization vector, the plumb line, is very important. So the way I've drawn this graph, for example, in this case, 1 and 2, you can show straight away you're going to have to be, I forget which way, where was my label? So 1 and 2 are Mobius, so these are parallel duplexes. So these are parallel duplexes, and the other 4 are anti-parallel duplexes. The way you see that, I haven't got the slides, is you just start walking around. You start walking around. So 1 is an odd duplex. So that means I finish at 6. So I start at, if I leave 6 to go towards 1, if it's an odd duplex, it means my strands going from here to here are going to have to cross. Okay, so it's the combinatorics of how things cross or uncross inside those vertices, which is a function of whether it's an odd or an even duplex, which describe this rigid, the edge ordering around each vertex of the rigid vertex graph. So I don't want to go into that in any detail. The point is, here's some pictures. So here's a fold, simple fold, 1 odd, 2 odd, 3 odd, 1, 2, 3. So 1, 2, 3, 1, 2, 3. There's no overbars, so these are all annular, which means these are all anti-parallel, and they're all odd parity. So that fold translates straight away to this polarized strand graph. And here's my plumb lines. So that means my double helix is running up vertically for all 3 vertices. Here's another fold, -1 odd, 2, 3, 1, 2, 3. And that means now I've taken my ribbon 1, and instead of making an anti-parallel duplex, I've made it parallel. So actually what that means is that I have to flip my polarization vector. So it runs the other way now, but it's the same graph, but it's got a new polarization. So what's interesting now is you can start to classify folds. So for example, this fold I can easily draw in this way, where the graph has got no edge crossings. And then I can put a skeleton inside the fold. So I color it black and white, checkerboard color it. The usual trick with knots is I can put a skeleton inside there, and I can think of this fold as being a string wrapped around that skeleton. So I have, in this case, because it's a one-colored skeleton, all the edges are inside the gray regions. It's always got a pair of strands locally, which are just winding around those edges. So it's got 1, 2, it's a theta graph essentially, and I'm just winding these edges around there. So that's that fold. If I take this fold, it's a bit more complicated because this has been flipped, the plumb line, that now the black-white colouring is quite different. And if I want to make it a one-coloured skeleton, in other words, all my skeleton edges are in the grey regions, I have to insert extra crossings in here, because remember, it's a rigid vertex graph, so I can't just flip edge orders. They're quite constrained. Think of the vertices as being a steel plate with 4 edges fixed, welded to it. So I can turn them and rotate them, but I can't just swap one to the other unless I cross them over each other. So that's a 3-crossing graph in that sense. But there's an easier way of drawing that fold where it has no crossings at all. So there's my 3 edge crossings. But now the skeleton is black and white. So the skeleton passes through black and white regions. And in fact, these exemplify 2 possible classes of folds. First class, simplest class, class A, would be where all my edges in my graph are uncrossed and the skeleton is one-coloured, or I could have all my edges uncrossed and the skeleton is 2-coloured. But there's a third class where no matter what you do, no matter how you embed that polarised strand graph, it contains edge crossings. So the first point is, do they have edge crossings or don't they? The second point is it one-coloured or 2-coloured? And any fold that I can build is either class A, class B, or class C. And this is actually quite a natural hierarchy in a sense, because as we'll see in a minute, these correspond in some sense to interesting RNA structures. So now let's try and look at these folds. So theoretically, any contracted fold can be very complex. So it might have junctions. So this would be a junction in here where we've got double helix, double helix, double helix meeting at a junction. It's going to have double-stranded domains, which are just the duplexes. And in this case, they could be parallel or anti-parallel. It's going to have single-stranded domains where there's no duplexing. And it's going to have also, in general, edge crossings, where there's no duplexing here. It's just forced by the graph combinatorics. So here's the result for if I have up to 6 ribbons. These are all possible folds which allow parallel or anti-parallel duplexes, which are contracted and which contain up to 6 ribbons. So here's the full list. So we've got these circular diagrams, we've got the fold label, and we've got the polarized strand graph for each fold. And then on the right column, these graph pictures, these are the skeletons around which this thing is winding. So sorry, when I said these are all folds, I was wrong. These are all A-type folds. So all of these fold very nicely around the skeleton. So I've locally got 2 edges along every edge of the skeleton, and along every edge of the skeleton, I've got a duplex. Now, in general, these folds are going to be knotted. So for example, if I take this simple fold, which is 1, 2, 3, 1, 2, 3, depending on the twist. So for example, this is odd, 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 but 3, 1, 3, or odd, 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 5, 3, 7. I'm going to get different knots. And in this case, these all happen to be pretzel knots. Now, the nice thing is that when I have A and B folds, because they have no crossings, if I embed, now, what I should say is I'm only talking about the simplest embeddings. Of course, I can take this polarized strand graph and I can always give it a pair of crossings anywhere by just taking my whole rigid vertex and flipping it one way or the other way. Okay, so I'm talking about the simplest embeddings, which are the embeddings which minimize the number of edge crossings in the embedding of the polarized strand graph. So if I have an A or B fold which has no crossings, that simplest embedding gives me a unique knot. If I have a C-type fold, I'm going to get multiple knots due to the edge crossing. So the idea is really that if I have an A or B type fold, the grand graph is topologically rigid, modulo this even number of things which I'm going to throw away for now. So the simplest embedding of the fold gives me a unique knot. And in that case, in these simpler cases where I have up to 4 ribbons, 4 annular ribbons, so there's no minus signs here. So these are all anti-parallel duplexes. So I'm going to call the twist A, B, C, or D. These just correspond to simple Conway rational links. So the first one would be A minus B, the product. This would be that this is the pretzel case, A plus B plus C. This is actually a composite knot. So the composite of two simple Conway product tangles. And here's a slightly more complicated one with 4 integer tangles. So now we can just write down a table where if I have a fold, for example, with this label, and if I know what its twists are, so this is twist 1 and this is twist 2, if it's got no crossings, they both have to be even, so they can only be 0, 2, 4, and so on. So you can keep on going forever. But if I just go through the combinatorics, if I have 0, 0, then it's what I'll call a pseudo knot or the unknot. 0, 2 is the unknot, 0, 4 is the unknot. But once I go to 2, 2, I get a knot, which is a trefoil, and so on. So you can classify these A or B type folds, which have no edge crossings, as knotted or unknotted, depending on the twists in each duplex. So depending on the number of half twists in each double helix, you're going to generate a knot or an unknot. But the point is that if I have edge crossings, so if it's a C-type fold, I can't draw it without edge crossings. And that gives me non-rigidity in a topological sense, because each of those crossings might be plus or minus 1. It doesn't matter to the polarized strand graph. It's just an embedding problem. So that means that I've got some flexibility in the knotting. So for example, if I had a fold here, 1 odd, 2 odd, 1, 1, 2, 1, 3, 1, for example, I could make that crossing over or under in any possible way. And I could get all these different knots from the same fold. So one way to think about it is if those edge crossings were randomly distributed, they could be equally likely to be plus 1 or minus 1. Then I have 4 possible combinations. 2 of them are knotted, 2 of them are unknotted. So you could say there's really a 50% chance of knotting in that fold. So now let's get on to RNA. I'm going to make the simplifying assumption. The point is I developed the machinery now to handle parallel duplexes and anti-parallel duplexes of arbitrary numbers of crossings. But now let's just zoom straight in on the most common form of RNA, single-stranded RNA, where it usually forms an anti-parallel. When it does form local duplexes, they're usually anti-parallel and they're usually right-handed. And the point here is that if I want to know what the twist is, plus or minus the actual structure, in general, 5 nucleotides contribute T equals 1. So the number of nucleotides in the duplex modulo that number 5, it's just over 5, in fact, gives you the twist. So it gives you an algebraic estimate of the twist. But of course, if you have a duplex with twist 2.3, in a topological sense, that's just twist 2. Okay, so you just go to the floor integer. So now, I showed you a table earlier where we had all contracted and uncontracted folds. Now, if we allow only annular folds, so we don't have any minuses up the top, so these are all annular ribbons, but we allow arbitrary twists, so that's what I call semi-flags. So the subscript is free, so I can have even or odd twists. Then here's now a table of the number of distinct labels. So this is really now the number of distinct folds where we don't care about odd or even twists as a function of the number of duplexes in the fold. So now it's become a bit more reasonable. As we go up to 5 duplexes, we get only 19 folds. So here's a cartoon taken from a very nice review from many years ago of single-stranded RNA. And the point they're making is here's the 5' end. So we're just following through the strand again. And we come out of the 3' end. Single-stranded RNA has a lot of interesting structure induced by secondary structure is really induced just by the nucleotide sequence, or you can also have tertiary structure which is induced by other interactions. So you can have, let me show you. For example, you could have single-stranded regions where you don't have any duplexing, and we've got those in our folds because they're just the edges of our polarized strand graph. You can have double-stranded regions where it's duplex. They're the vertices of our polarized strand graph. You can have stem loops, so it's just a hairpin bend. We don't care about them. They're gone. They're gone because they correspond to those free arcs around the boundary that we just pull off. You can have bulges. We're going to remove them, so we just pull them out and just think of that as a single duplex. This is the same thing. We can have what they call mesojunctions, where now the orientation of the three, we've got three duplexes coming into this junction, and two of them are the same orientation, but this one has been flipped. And that is actually a two-coloured embedding. So when we have a two-coloured embedding, we have mesojunctions. When we have a one-coloured embedding, we have so-called conventional junctions, which are more common in biology and easier to make and so on. These ones are quite interesting because people sometimes talk about this as being tertiary structure, but in my sense, I can classify this within this fold description. So these are so-called kissing loops because the idea is that these are non-adjacent regions of the strand which are hydrogen bonding and duplexing. So they're allowed. And then lastly, we have these so-called pseudo knots. Now, the interesting thing about a pseudo knot, and I'll define it in a minute, is that all of my non-trivial folds are actually pseudo knotted. The definition of a pseudo knot is really just that the fold diagram has crossings. That's essentially it. They call it a pseudo knot because they don't believe that these things are knotted in active RNA. So they're pseudo knotted. So they have this almost knotted property, not quite knotted. So now here's a list of all the contracted folds of any type, A, B, or C, only with antiparallel duplexes and up to 4 ribbons. So this is type B, this is type A, this is type B, B, C, C. So these two have edge crossings no matter what I do. These 4 have no edge crossings in their simplest embedding. And the nicest fold of all is this type A fold because this has so-called conventional junctions everywhere. This has mesojunctions, this has mesojunctions, and so on. So here's the strand graph pictures. Now notice that the zero crossing, this is what I call the semi-flag diagram, right? So I haven't put any parities, any twists here. But if I want to have zero crossings, I have to make both duplex 1 and 2, in this case, even parity. And these are odd, 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 and so on. So these are the actual fully flagged diagrams with zero crossings. And then if you go to 5 ribbons, you get a much longer list. So here's a pseudo-knotted fold, a cartoon again. We start at 5', where's it gone? 5 primed, then you move. Where is it? I'm lost. I've lost the orientation myself. I can't see the picture. 5. It's a continuous strand, and basically, I don't want to waste time on it, but you go blue, red, blue, red. So you go blue, red. Oh yeah, there it is. Blue, red, then you come back around, then you go back through blue, and then you have this loop, then you go back through red. Okay, so the linear diagram, but that's the simplest pseudo-knotted fold in RNA. It's called an H-type fold, and it's very common in viruses especially. So that fold diagram is really just my simplest fold diagram. So now I have to do one thing more because instead of a circular diagram, I've got to cut it open. So depending on where I cut one of my circular diagrams open, I can get different linear diagrams. So here's the same list, just giving you all the linear diagrams with up to 4 ribbons in the linear fold. Now the point is, if I have an even parity duplex, I can cut it in the middle of the duplex, and that can be 2 odd parities or 2 evens. So I can bisect inside a duplex as well, and that gives me all these different diagrams. Now, if you look at this list, this is just my mathematical list ranked. In fact, the first one is the H fold, which is very common. The next 3 are all what biologists call K folds. Sorry, the next 2 are K folds. These are also reasonably common. The next one is an L fold, which is slightly less common but exists. And then these physicists that I referred to in the opening slide discuss also this M fold, which still hasn't been seen to my knowledge. So, but my point here is that I would expect them to come out in biology or in material science, if you like, in this sort of ordering. Now let's look at the in detail an H pseudoknot, and then I'll finish. And this is the pseudoknot actually from the SARS-CoV-2, which is a virus that causes COVID. So this is very relevant actually, because some people are thinking, I'll give you a reference in a minute, that we should be perhaps an interesting target to inactivate, because all these coronaviruses, well, many nasty viruses, as I said, dengue, Zika, SARS, COVID, have this characteristic pseudo-knotted fold in them. So people are suggesting maybe we should be targeting trying to attack that pseudo-knotted fold in some way instead of coding for the proteins and so on. So this is really attacking the RNA, not using the RNA to produce proteins in the body to give you antibody resistance, but attacking the RNA inside the virus point blank. So it's an interesting idea. So here's a map of a cartoon of the SARS. The SARS thing is actually 30,000. The genome is actually 30,000 nucleotides. So this is just a small region inside there, about 80 nucleotides. And this is just taking a small bit inside there. Everywhere else is just other stuff that I'm not going to talk about. But this is the bit that's interesting. So here's the 5' end. So you go along here and through blue, a loop, then through orange, then blue, then purple, then a hairpin, then purple, then a loop, then orange, then out. So that fold I can draw very crudely metrically by assuming that every nucleotide is equally spaced along the length in this way. So this is a sort of metric realization of that part of the fold. It's actually nucleotide number, that's starting at 13,418 and going up to that plus 80. So if I look at that fold and calculate the number of nucleotides in each helix, I can work out roughly my twist just from this divide by 5 more or less rule, and then collapse it down to the lowest integer. So that's essentially, in my language, contracted to 1, 1, 2, 1, 1, 2, because here's one of these, this is one of these loops that I throw away because that's got nothing. It's a free annular loop, so get rid of it. So this is just 1, 2, sorry, green, red, green, red. So that's 1, 2, 1, 2. So that's actually a C-type fold because it has odd parities. And because it has odd parities, I've got 2 crossings in there. So depending on how I choose my crossings, they could both be -1, they could be -1, 1 or 1, 1. So these are the crossings in the embedding of the polarized strand graph edges. These are not crossings inside the duplex. These are in the single-stranded regions. So I've got 3 possibilities modulo symmetry, 2 of which are unknotted, or now this is why we call them pseudo knots. These RNA people call these unknotted configurations pseudo knotted. So if it's unknotted and it has a cross diagram, it's pseudo knotted. So I have pseudo knot, knot. So in that sense, there's a sort of third chance of that thing being genuinely knotted. Now that's quite interesting actually, because surely that's not what happens in the real virus. Surely that virus is 100% pseudo knotted, because if it were knotted, you can't translate it. Presumably, you can't use the ribosomal machinery to make what you need to make, the proteins and so on, to build the new virus while that thing is knotted. But actually, the pseudo-knotting has a very interesting function because it's almost a knot. It's a bit like a tangled bit of string. As you fit one very nice, well, it's almost certainly the fact, is that as you feed it into the ribosome to translate it into a protein, that acts as a topological obstacle. So it's pulling. So it's reading that branch of RNA encoding a protein. Then it pulls it and breaks that tangle, and all of a sudden it's a sudden shift. So it can actually read 2 possible combinations from the same strand of RNA because of this pseudo knotting. So it's a very clever way viruses have evolved to give you multiple protein readouts from RNA by using that pseudo knot. So that pseudo knot is very important. If you could get rid of it, or if you can mess it up in another way, you're going to mess up the protein build. But the point about my analysis is really just, surely this is not knotted. So there must be something else which is disfavoring these +1, +1, +1, sorry, -1, -1, -1, +1 crossings. Sorry, wrong way around. We want something which disfavors one of the 3 combinations because that's knotted. We're happy to have these. Well, the virus is happy to have it, we're not, but it's not happy to have that. So I believe that what's going on there is there must be more tertiary information. And we can actually encode tertiary information with this as well, but I haven't got time to talk about that. So just to finish the opening slide, I talked about this problem of trying to understand knots in RNA. And they finished their paper by saying a most interesting research avenue would be to extend the scope of RNA structure prediction to distinguish different forms of entanglement. So I've gone some way towards understanding that, but the answer is that the answer to their question is that it's really quite complicated. In fact, any fold could be realized as an unknotted fold if I put enough crossings, edge crossings in the graph embedding and tune their plus or minus 1 suitably. So thank you. That's really all I want to say.
Thanks, Stephen. That was really interesting. Any questions, Stephen? So as before, if you want to type in the chat, if you don't want to, or you can't, shout out. But if you can shout, feel free to.
Herbert, I think Herbert needs to, I owe you a discussion, Herbert. Sorry, are you still dissatisfied?
I think it was a misunderstanding of a definition on my side. What you call a duplex is you twist it up and there's lots of crossings. I thought at every crossing there is hydrogen bonds, but actually all along this twisting structure is hydrogen bonds.
Yes.
So you have to twist. You cannot just alternate.
Yes.
Yeah.
Yeah. I mean, you could alternate, but that would be a separate duplex.
Yeah. Right. Right.
And that can happen, in fact.
Can I ask a somewhat different question? So I have this memory, HIV is a virus, and one of the standard treatments for that virus is you inhibit a protease because it can only replicate if this other protein will come in and cut what's going on in the replication, and it will come apart in order to, and then go back together. So the idea of cutting and then re-gluing as a functional thing in a virus is certainly there. It may not be RNA, that's what I can't remember. But the inhibitor is to stop the cutting because it's tangled and it needs to untangle and get through something. So it's fairly common to actually cut and re-glue things in biology.
Yeah, that's right. I mean, there's certainly true that these topoisomerases that do this cutting are active in work on RNA. That's been proven, right? So we know, you're right. I agree with you. In fact, when you mention HIV is another one of these viruses that has this pseudo knot fold in there. I think it's the H type from memory, but yeah. I mean, that's an interesting point. That's sort of what made me, when I first started on this, that paper where people said, well, a lot of people said, well, of course, RNA can't knot because, you know, it would be such a mess with the whole ribosomal translation work. But your observation, I agree with, given that you have topoisomerases and given that they're active on RNA, presumably that means it has to do, you do have this complication in RNA where things can be knotted. It has to unknot it to do some work.
Right, and if you can stop that, you can stop the replication.
But so far, I haven't found a virus RNA which, in my theory, should be genuinely knotted. Or I haven't found one which is going to be 100% knotted. I found some which are 50%, 30%. So, for example, the idea for me would be if we could switch off, another strategy would be switch off some of those tertiary interactions, which are favouring some of those edge crossings over others, and you're going to push it into the knot regime and then it's finished. So I'm not a doctor or a medical researcher, but these ideas I think are probably relevant.